Steve. Thank you all for being here to hear about Avery Therapeutics. The technology in Avery is a platform technology for tissue engineering. It came out of the lab of Dr. Stephen Goldman and Drs. Jordan Lancaster at the Southern Arizona VA and the University of Arizona. We have also recently brought on George Dunbar as our executive chairman. So our platform works at solving the underlying problem of cell death, and that cell death could be caused by, for example, a heart attack or a trauma, but ultimately that cell death leads to a loss in function. We are engineering tissues that are allogeneic, cryopreserved, scalable, and that makes them cost-effective. We implant these at the site of cell death in order to regenerate tissue and ultimately restore function. The platform that we use includes therapeutic cells that are tailored to the indication, combined with fibroblasts, which are a general cell of the body that result in extracellular matrix generation, providing support for the therapeutic cells, and are also highly angiogenic, so they grow blood vessels in ischemic areas. We engineer these cells into a tissue using a bioabsorbable scaffold. The scaffold provides the advantage of robust handling of that tissue so that it can be deployed minimally invasively, it can be sutured, and because it's a sheet-like tissue, we can cover the entire area of injury. So our first product in development is an engineered heart tissue, which we're calling myocardia. It uses iPS-derived cardiomyocytes as the therapeutic cell. This is a video of the tissue, which beats synchronously and spontaneously in vitro, so this is without any electrical input. When we implant this onto the surface of the heart, we suture it in place, it integrates into the heart, and it electrically matches the pacing from the SA node of the heart. So it improves the electrical conduction through the heart, and it regrows blood vessels and heart muscle. The histology images that I'm showing you here are from a rat model of heart failure. The top image is a rat that has chronic heart failure. We achieved this with a permanent ligation of the coronary artery in the rat. You can see that the ventricle, this is a cross-section of the left ventricle, is dilated, and the blue section there is the scar tissue, the ischemic area on the heart that has become thinned out. There's very little surviving muscle in that area. The bottom histology image is a left ventricle cross-section from an animal that's been treated with our technology. You can see that the ventricle has remodeled. It's smaller in diameter, and there's increased thickness of the area where that tissue has been implanted. And we actually see new blood vessels and new muscle in that entire region where we've implanted the tissue. We've used MRI, echo, and hemodynamics to show both systolic and functional benefit in these animals. We've tested in hundreds of rats, and we've done pilot studies in swine. We have an ongoing study in swine looking at 6 months post-treatment. And these animals show benefits that would be resulting in an improvement in quality of life for patients with heart failure. We believe that we can use a single dose application for these benefits. And because we're using an allogeneic cryopreserved product, it can be stored frozen and then thawed and implanted when needed. Our IP covers the composition of this heart tissue as well as other tissues for other muscle and enzymatic tissues. The funding for the company so far has come from one angel round in which we raised $575,000. We've also had a large amount of grant funds. These funds have gone to the rat and swine efficacy studies, which have used all human iPS cells, as well as development of manufacturing techniques that are highly scalable. We will be raising a Series A. The $5 million will get us to IND. So that $5 million will be used to do the phase 1 GMP manufacturing and the IND enabling study. And then we'll conduct a phase 1 clinical trial in patients with class 2 to class 4 heart failure.
Great, thank you. Thank you, Jen. So maybe to start, as an early stage company, obviously you're targeting heart failure as an initial indication. You mentioned some of the other possibilities for the technology, other muscle conditions, other cardiac conditions, DCM you had on there. So how do you make that decision about what to go after initially and what to prioritize? And speaking to heart failure specifically, what are some of the advantages you see for you there?
Yeah, so we're starting in heart failure because it's a very big problem. There's 26 million people worldwide with heart failure. We have very good access to that patient population. Our chief medical officer has been a cardiologist for over 40 years. He sees these patients. He sees the lack of therapies that are there for them. There's a real unmet need for patients, particularly that are in the class 2 to class 3 heart failure, where their symptoms are progressing and there's no effective therapy. So heart failure also is an advantage for us to go after because of the team that we've built around the cardiologists, the cardiac surgeons. For all of our swine implants, we've used cardiac surgeons to do those implants. So they've handled the technology. They've given us feedback that has allowed us to improve the technology so that it's easily deployable. And the models for heart failure are very good models. The rat model is really a worst case scenario model with a large infarct. We're able to see benefit there. So it's a natural progression for us for our first technology, our first product.
That's great. And you mentioned the preclinical models that you're using. So this is not necessarily a condition where you need to do non-human primate studies. It seems that the rat and swine are the best models here to get you ready to move into the clinic.
Right. So the rat model is very good because, as I said, you can get a very severe infarct, and we're able to see huge benefits with our technology in that rat model. For the scalability to the clinical size, we're using the mini swine model, and we've been able to demonstrate that we can scale up the technology to a human-sized graft and use minimally invasive surgical methods to deploy that technology.
So in addition to the graft itself moving from the preclinical models to the clinic, obviously the surgery changes. How will that play out? Is it going to be a period of trial and error in terms of the surgical technique, or is this a very straightforward procedure to deliver the graft itself? You mentioned suturing it on. Maybe expand on that a little bit.
The process is actually quite simple. A trained cardiac surgeon is used to sewing things onto the heart and performing minimally invasive surgeries. So we've used cardiac surgeons for our implants in swine. We've also implanted in human cadavers. In the swine on a beating heart, it takes less than 10 minutes to implant the technology. And in a human cadaver, we've demonstrated with minimally invasive techniques that with minimal additional training, the surgeon is able to perform this implant successfully.
That's great. And focusing on the graft itself, obviously the cell sheet or the scaffold approach different than a cell suspension or a cell injection approach. Maybe just help us understand the differences there and why you think the scaffold approach in particular in this indication is ideal.
Yeah, so the scaffold approach and the tissue approach is really the key advantage to our technology, and it's something that we really think is necessary for the heart, that in order to get that functional improvement, you need to provide something that is functional. So with the intact tissue, we're able to bridge over the entire infarcted region and treat that entire area. we're also able to induce blood vessel growth into that infarcted region so that we have the nutrient support to keep those cells alive and to regenerate that tissue. That's an important thing that you're not able to see with the cell injection technologies because they're in focal areas. They're not over that entire area. You're not able to get the electrical benefit that we see, which is increased voltage amplitudes, decreased time for signal propagation across the infarcted area. In addition, the value of the tissue approach is that we know the exact dose that we're putting on there, and there's not cell loss, whereas in the injection approach, it's hard to know how many cells are actually staying in place. When they're in an intact tissue that's applied to the surface, they're not going to be squeezed out as the heart is pumping. There's nowhere for them to go.
Great. And one thing that you mentioned was the ability to, you can freeze down these grafts and store them. Maybe you could just comment a bit on the process of getting from point A to the final graft. What is your ability? What is your capacity to store these, and at what scale can you do this as you think about growing into eventually commercialization?
Yeah, so the ability to cryopreserve our cardiac tissue is another key advantage to our technology. It's important because we're using an allogeneic approach, a single donor approach, and with the allogeneic cryopreserved approach, we can make these in bulk and store them. We believe we can store them for at least a year on liquid nitrogen temperatures, and they can be stored in the hospital frozen and thawed and used when needed. So that makes it highly cost effective because we can manufacture them in bulk. And one of the grants that we got was an NIH grant specifically focused on closed system manufacturing. In that grant, we demonstrated that we can manufacture these tissues in a closed bioreactor system where each dose is manufactured in a single bag that then becomes the internal shipping compartment. So there's no user handling of the tissue itself. It stays in that bag the entire manufacturing process all the way to when it's ripped open and implanted. That helps keep the sterility, and also we can manufacture hundreds or thousands of those bags at one time, so then when it comes to meeting that market need of millions of patients, we'll be able to scale up our production.
Great, and given the allogeneic approach, the iPSC approach, what things do you think about when you select donor tissue? How much variability is there between donor tissue you might choose to select, and what are the criteria that you look for there as you think about optimizing, obviously, the master cell bank, I guess?
Yeah, so for us, we're planning to use a single donor for our IPS bank, so a single IPS bank, and our real focus is just a young, healthy donor. We've tested iPS cells from multiple donors. We've tested multiple cardiac differentiation protocols, and we've seen safety and efficacy in all of the cells that we've tested. So it's really, those have been young, healthy donors, and that's what we're sticking with.
Okay, great. So maybe just in the time we have left, beyond heart failure, we did talk about some of the other indications that are possibilities. Dilated cardiomyopathy for one. Maybe you just wanted to comment on the rationale there, other cardiac conditions that you'd be looking to pursue in the future, and why don't we stop there, and if we have time, maybe we can get into non-cardiac conditions.
Great, yeah, so this really is a platform, and we're looking for partners that are developing other cell types that we can use in this tissue platform. The dilated cardiomyopathy is a natural progression for us because we see remodeling of the ventricle, and that's what a patient with dilated cardiomyopathy has, a dilated ventricle. Restoring that to a normal-sized ventricle would improve the health of that patient, so that's a natural next step. We have other cardiac conditions that we're looking into related to the improved electrical conduction that we see with this technology, as well as the non-cardiac applications that we're seeking partners for.
Great. And what are the key steps for you guys over the next year or two? Will you be in the clinic? What is the timing there? And just anything else that would be a big milestone for the company?
So the next step is doing our phase 1 GMP compliant manufacturing and our IND enabling study for the heart failure IND. And we'll be submitting that IND mid next year, starting our phase 1 clinical trial in 2019.
Okay, great. Thank you, Jen.